Well, good afternoon, everybody who's in the audience and also on Zoom. So today's speaker, Andrew Persichetti, our recent K-99 grant recipient, first joined the lab in 2018 as a postdoc after getting his degree from Emory University. And he joined at a particularly opportune time because Renzo Huber, who's right there, and Peter Bandettini and colleagues had just developed a non-mold dependent neuroimaging method that would allow us for the very first time to evaluate neural functioning at the level of cortical depth. That is, at the level of the laminar. And Andrew quickly realized the potential of this method for addressing a whole host of really interesting questions about the architecture of cognition related to feed-forward and feedback processes that we know are dependent on the laminar and their interaction, which is exactly what he's going to be talking to us right now. So there's his title, Dissociating Feed-Forward and Feedback Information Across Cortical Layers in Humans. Andrew, please.
Can everybody hear me? Good, yeah? What? Hold on, this is not, I'm not seeing that on my screen, but no. What's that? Oh, maybe here. Oh, here we go. Got it. Okay, all right. Thanks, Alex, for that nice introduction. And thanks for the organizing committee for inviting me to give this talk, and all of you for being here. So today I'm going to tell you about some work that we've been doing using fMRI to look at the laminar organization of human cortex. All right, well, before I do that, let me take a step back and just provide a little bit of context. So fMRI has advanced cognitive neuroscience in many significant ways. So we've been able to characterize whole brain functional networks. We've also been able to look at how information is processed along hierarchies. So for example, the dorsal and ventral visual pathways, also known as the vision for what or where and vision for what pathways, first proposed by Leslie Underleiter and Mort Mishkin over 40 years ago. And all along this hierarchy, for example, we're able to look at particular regions and characterize the response tuning of these regions. So for example, face tuning in the ventral visual stream starts with low-level properties in V1, then goes to category selectivity, and then down to coding of individual identities. And we're able to do this in typical and clinical populations. And we're able to look at this across the lifespan with fMRI. However, obviously, lots of questions still remain. Two of them that are of particular interest for layer fMRI are first, what does it mean for processes and representations to overlap in a region of cortex? So for example, there's lots of evidence that when I look at something, for like a dog, or if I simply imagine it without visual input, there's both processes will evoke overlapping responses in V1, in primary visual cortex. However, these are fundamentally different processes. So we need to understand how we can differentiate them across the cortex, specific regions. The second question is, what is the direction of the flow of information between regions of a network? So we know brain systems are highly recurrent. So we need to find a good way of understanding how feed-forward sensory information and feedback information from downstream cortical regions propagate throughout the net. So one way to try to do that is by using, by looking at the cortical layers. So looking at this dimension of cortical depth, that's a little blurry, in the cortex. So we know that the cortex is organized into layers based on cytoarchitecture. And we also know a bit about the functional organization across these layers, particularly in primary sensory and motor regions. So for example, in V1, we have a simple model here where feed-forward information from the eyes goes into the middle layer.
You've already, gone up like the brother.
Okay. Somebody mute. Whereas feedback information from other cortical regions feeds back into the superficial and deep layers. So just knowing the organization of this circuit, we can start to answer these two questions. We can understand, we can map out different processes. So for example, actually seeing something versus simply imagining it. And we can also start to make inferences about the direction of information. If we know this information is feed-forward information or more feedback goal-oriented, for example, types of information. So today I'm going to tell you about how we're using layer fMRI to measure feed-forward and feedback information across the cortical layers. I'm going to start by just giving a brief background for layer fMRI. And then I'm going to talk about an experiment where we use the layer fMRI in primary motor cortex. And I'll explain in a minute why I'm pivoting from talking about visual systems to the motor cortex in a little bit. And then I'll bring it back to the visual system and talk about an ongoing project where we're using layer fMRI in the ventral visual stream. So first, just for background, the way we typically use fMRI, we typically look at we look at the bold signal, which is measuring changes in blood oxygenation levels as blood flows into a region in response to neural activity. So a very basic example of an fMRI experiment might be to have people tapping their fingers together and asking where in the brain is this happening. And among other places, you'll find activation in primary motor cortex, the hand-selective region of primary motor cortex. And while this tells you about the location of the information of where this process is happening, that it tells you that M1 is in an active state when someone is tapping their fingers. It doesn't tell you about the processes happening during the finger tapping. So again, we can look at layers and get some information about this. We know that M1 is organized in a simple input-output circuit, where cortical input and communication with other cortical regions feeds into the superficial layers, and while the deep layers send signal out to the spinal cord to initiate body movements. And for the remainder of the talk, I'm not going to talk about specific cortical layers. I'm just going to talk in general terms of superficial, middle, and deep layers. So if we're able to measure layer-specific responses, then we can get at the processing that's happening in M1 and not just that it's active during a task like finger tapping. However, a problem for using looking at the layers with fMRI is that we're not directly measuring neural activity. We're measuring changes, vascular changes. So we rely on this neurovascular coupling between the vessels carrying the blood and the neurons that they're feeding. And a big problem here, specifically for the bold signal, is that it's dominated by these large veins on the surface of the cortex and these large ascending and descending veins that are not specific to any layer. So when we measure the bold signal across layers, we'll just get a very large superficial bias. And this the superficial bias is obfuscating or potentially obfuscating all the activity happening outside of the superficial layers. Oh, and then, so I'm going to show more of these response profiles plots, and when I do, superficial is always on the left, and it goes to the deep layers on the right, just to orient you here. So what we've been doing, instead of trying to measure the bold signal to look at layers using fMRI, we're using a method called VASO, or vascular space occupancy, which Alex briefly mentioned in that introduction. And real quick, just lots of the developing of VASO has been done by Renzo Huber, who's an MR physicist in Peter Bandantini's lab in Building 10 in the imaging facility. And for the rest of this talk, lots of this work hinges on having Renzo here and his technical expertise. So the nice thing about VASO is instead of measuring the oxygenated signal that's coming from these large non-specific veins, it's measuring inflow effects so when the blood flows in, not only is it oxygenated, but it's also causing the blood vessels to dilate, they get larger. And this is the mechanism that VASO is measuring. So, and the nice thing about that is this dilation signal comes from these smaller layer-specific blood vessels found across the cortical depth. So when we plot the VASO signal now to a finger tapping path, we'll get a double peak response that's reflecting this input-output circuit that I just described. So with VASO in hand now, we did an experiment in M1, building off of this double peak response that Renzo and others had already shown in M1 in response to tapping. And we wanted to ask two questions. First, what does it mean for imagined and executed actions to overlap in M1? So just like I was talking about in V1, there's lots of evidence that executing an action or simply imagining that both evoke overlapping activity in primary motor cortex. And the second question is, how does imagining an action result in subsequently improved performance? So there's also lots of evidence that if you simply imagine doing something like maybe playing a piano, when you subsequently play the piano, you'll do it a little bit better. And this concept has been adopted large scale. Lots of professional athletes even use this type of visualization as part of their training. And before moving forward, all of this work is the product of my co-authors and especially my lab mate, Jason Avery, and again, Renzo. So before telling you about the experiment, let me give you that explanation about why I've pivoted now to primary motor cortex. And there's three practical reasons for doing so. The first is it's the thickest part of human cortex. It's on average between 4 and 5 millimeters thick. So we have lots of real estate to be able to measure, to dissociate layer responses. The second reason, as I just described, Renzo and others have already shown that VASO works in motor cortex and that you do get this double peaked response when you're actually executing an action, specifically tapping your fingers together. And the third issue has to do with, I think, just fMRI in general. So layer fMRI requires high resolution, sub-millimeter resolution, so very small voxels. And whenever you make your voxels very small, you make your signal very noisy. So whenever you can get some of that back, it's always a good thing. So a nice thing about so to do that in this experiment, we've made our voxels thicker in the slice direction. And I'll explain why M1 was important for that. So M1 is positioned, this is a little blurry, but it's often called the hand knob, the hand selective region. It's this knobby little guy right here. And so what we can do with our field of view is position it perpendicular to this hand knob so that we're looking down on it, essentially, so that our thicker slices are cutting across the gyrus, but the sub-millimeter resolution is still in plane. So our slices are thicker, but if you look within any one of those slices, we have high resolution. And you can see here, you can easily see the segmentation of the cortical ribbon here. And this is just a close-up view where we can easily go in there now and identify the white matter, gray matter boundary and the cerebral spinal fluid and the gray matter boundary. So what does it mean for imagined and executed actions to overlap? So to answer this question, we had participants complete 3 conditions. First, we just had them simply tap their fingers together. And we predict that we're going to get this double peaked response that's been shown before. We also had them keep their hands still while simply imagining tapping their fingers together. And we predict here that we're going to find activity in the superficial but not the deep layers. And in a third condition, we had them keep their hands still and simply just wiggle their toes. And this is just kind of an active baseline. We don't expect hand selective M1 to be active when someone's wiggling their toes. So we should just see activation fluctuating around 0 across the layers. And we did this in a slow event-related design. Our TRs were relatively long, a little bit over 3 seconds. So we can't have, our stimulus presentation needs to be a little bit slow too. And we presented these conditions in a counterbalanced way so that we could measure both the direct effects of each condition and also the carry effects of the prior condition on the present one. So in plain language, how is this tapping condition affected by the preceding imagined tapping? And that'll become important when I get to question 2. So before looking at the layers, we need to identify them in the cortex. And to do that, we first anatomically found the hand selective ROI, which as I told you before, is anatomically easy to identify. It's called the hand knob. It has a knobby structure that's pretty easy to see. And then we used our tapping condition simply as a functional localizer. So this is activation in one subject. And you can see in this region of M1, you can see this clear double peaked response along a large swath of it. So we identify it, and then we go and we draw our layers across the cortex where this double peaked response is. And here I'm just showing you the double peaked response so you can kind of orient yourself to it and the actual brain data. So when I show you these response profiles, it's actually relating to these double peaked responses across cortex. Okay, so now that we have a hand ROI that we can look in across the layers in, we scanned 11 subjects. Oh, and to be clear, we defined this hand ROI individually in each person. And again, we have our functional localizer. We have a double peaked response in each person. Again, this is non-independent. We use it as a localizer. But now we can ask in this hand selective region, what's the response to imagined tapping and toe wiggling? And as we predicted, we see activation during the imagined tapping task in the superficial layers, but not in the deep. And next, to quantify this effect, we again use the functional localizer to identify the peak responses in the superficial and deep layer so we could average over them and compare our conditions. And indeed, we find a significant difference in the superficial layers between imagined tapping and toe wiggling, but not in the deep layers. And critically, there's a significant interaction between the superficial and deep layers. So we showed here that even though we get activation both for executed and imagined tappings in M1, these processes are dissociable across the layers. So next, we wanted to know if we could look at the circuit and get an idea of how imaginary interaction might result in improved performance. And to do that, we looked at repetition effects. And so to be clear here, we're taking all of the tapping blocks and we're separating them based on the condition that preceded them. So repeat conditions where it's preceded by tapping, preceded by imagining, tapping preceded by toe wiggling. And a commonly reported type of repetition effect is called repetition suppression. And repetition suppression is found in lots of regions of the brain, it's not specific to M1. But the way that it works is that you'll get a significant reduction in brain response when you repeat a condition relative to when that condition is preceded by a different condition. So we expect to see that from our repeated and the toe wiggling condition. And then we can ask, do we see the same type of repetition effect for the tapping condition when it's preceded by imagined tapping? And so here's just those data shown plotted across the layer profile. And we also quantified this in the same way that I just described by averaging across the peaks. And we do indeed find repetition suppression, like I just described, for the repeat condition relative to the completely different. And we also see a reduction in response to the imagined tapping condition as well. And so we think this repetition suppression in the superficial layers might reflect more synchronization with other cortical regions that are involved in motor function and motor planning. And that type of synchronization would likely lead to more efficient processing. Now in the deep layer, we can ask the same question. And interestingly, we find the exact opposite pattern of response. We found a repetition enhancement effect for both the repeated condition and the imagined tapping condition. And repetition enhancement, less commonly reported than suppression, but there is a literature on it. And we think that this repetition enhancement in the deep layer might reflect kind of a gain increase in the signal to send a more robust output to the spinal cord, thus resulting in higher precision. And so to quickly summarize here, first I showed you that while we get activation for both imagined and executed tapping in M1, these processes are dissociable across the layers. And then we also I showed you some repetition effects that we think are really exciting because we think they might explain how imagining improves subsequent performance, and that's by sharpening the cortical input and enhancing the motor output. And we're now answering whether or not this can be linked to behavior, and we're doing that in several ways. One way is through a collaboration looking at short-term motor learning effects in this area with Leo Cohen's lab in NIMDS. Okay. So now let me circle back to where we started from in the visual system, and specifically in the ventral visual stream. So while we have a pretty good idea of the feed-forward and feedback organization across layers in V1, we really have no idea or what this layer organization looks like going downstream in higher level visual regions. So in this ongoing experiment that's still in its early stages, we want to do two things. We first want to develop layer fMRI protocols, a protocol that works in ventral occipital temporal cortex. Lots of layer work, vaso included, is done in primary motor and primary sensory cortices, and mostly in M1 and V1. And second, we want to establish the organization of the laminar circuitry and regions of the ventral visual pathway. And before I continue, all of the progress made in this experiment are the results of a team effort by my lab mates, Sam and Taylor, that have done excellent work helping to develop the data processing and data analysis pipelines that we use. So the first thing we have to do to get this, to develop this protocol is overcome some quite significant challenges for using layer fMRI in the ventral visual stream. So some of these problems are not specific to VASO or layer fMRI. It's just high resolution EPI often results in some messy data. So but with VASO, they tend to be particularly bad. So when we first started this, we had massive distortions and artifacts all across the brain, and importantly, in the regions we care about. And also, quite significant drop in our signal in the regions we care about. And through just endless tinkering for the better part of a year and a half, I'm happy to say that these are now just before pictures. And we've made great progress in getting some pretty nice VASO images out of across the brain and importantly in the ventral visual stream. And we've also been able to boost our TSNR quite significantly. So the other two problems are more specific to layer fMRI, and they have to do with registration and segmentation, which are usually trivial in fMRI experiments. At least for me, I never gave it too much thought. However, with VASO, we have a difficult problem of getting our high resolution, nice distortion-free structural image to register to these distorted and a bit noisier VASO images. And the other added problem here is that our images are nowhere near full field of view. We're not collecting whole brain. We're collecting a little slice of the brain, a few centimeters. And to solve this registration problem, I have to thank Paul Taylor and Daniel Glenn from the AFNI group. They were pivotal in getting this to work. And then the other issue is just the segmentation. So once we have this registered, we need to be able to draw segment the cortical ribbon so we can then identify the layers across the depth. And so here, again, lots of help from other people, Farouk Golban at Maastricht and Tyler Morgan, a staff scientist in Peter Bandettini's lab, has helped us a ton in just being able to figure out pipelines so that we can segment this more or less by hand. So overcoming those challenges and the endless headaches of a year and a half, we can now start to ask questions about the layer profiles in the ventral visual stream. And so in this first project, we just want to ask how do imagined and viewed pictures overlap in the ventral visual stream. And specifically, we're going to look at two well-known areas, the fusiform face area, or FFA, and the parahippocampal place area, or PPA. And we're going to use V1's layer profile as a guide and ask, are we going to get the same layer profile as you go downstream? We'll assume yes, but we could be proved wrong. And so the way that we're going to isolate this feed forward and feedback information across the circuit, again, is just showing pictures of faces and places or having people simply imagine the faces and places. And the paradigm that we've been using so far, and this is still, again, early stages, we've collected about 4 participants, but they're still can be considered pilot data. We have a perception condition. They're looking at famous faces and famous places. So we have a perception condition where they'll see the name of a person, in this case, Barack Obama, and immediately followed by a picture of him. And and the other condition is an imagery condition where they will see the name Barack Obama, but now they'll just see a white empty picture frame, and they're asked to fill the picture frame with a portrait of this person. And we did the same thing for the places. So as I told you, this is still in the very early stages. So I don't have any data to show you from this, but I do have some preliminary data that I'm really excited about, highly relevant to this, that I'll just go ahead and show. So keep in mind, this is an N of 1, but we have seen this in a few other people since. I did not have time to incorporate it into the talk. But in this person, we just showed them 3 runs of just non-famous, famous people and scenes. And they saw 3 runs, about 11 minutes each. And we used a basic regions of interest design here, where we functionally localized the FFA and PPA based on the contrast of faces versus places. So faces greater than places for the FFA, and the opposite in the PPA. So then we can use our other 2 runs to look within the layers here. So we functionally localize. We then, as I described a minute ago, we hand segment the cortical ribbon there, and we can draw layers across it. And now we can look at our independent other 2 runs of data to see what the layer profile looks like in these 2 regions. And again, we're asking, does it look like V1? Or does the layer profile change as you go downstream? So before I show you the vaso data here, though, I want to go back to what I was telling you about the bold signal. Remember, I told you that the bold signal has a huge superficial bias. And also the cool thing that I didn't describe to you about vaso is the sequence works by collecting interleaved acquisitions of bold and vaso or null images. So we have bold data along with our vaso data in the same experiment. So that's pretty fun. And so I can show you here 2 things. First, there's a huge superficial bias in both of our regions, like I described earlier. And the other nice thing to show here is in our independent data here, we get the same specificity, right? There's a greater response to faces and places in the FFA, and the opposite pattern in the PPA. So now the fun and exciting reveal. With the vaso data, we are in fact seeing evidence that the feed-forward information is going to the middle layers, similar to in V1. So in contrast to this big superficial bias, we're now seeing, again, category selectivity, which is great. We're seeing that the peak of activation with the vaso data is being pushed more into the middle layers. And again, this is just one person. We're seeing similar results in other people. And stay tuned to see if we can get a nice imagery effect to show the feedback, the complementary feedback parts of the circuit. So to summarize, I showed you how we're using high resolution fMRI and a method called vaso to measure feed forward and feedback information across cortical layers. And I showed you some work showing that we can dissociate imagined and executed actions across layers of primary motor cortex, and that the pattern of repetition effects in this region suggests that imagery may improve behavior by sharpening cortical input and enhancing motor output. And then I finished by showing you some preliminary data going beyond primary sensory motor cortices and looking at what the layer organization is in high-level visual cortex in the ventral visual stream. And with that, I'd just like to thank everybody on this slide that's been critical to all the work that I've just presented, and a special thanks to the National Eye Institute for awarding me a K99 to pursue this work. And with that, I can take questions, and thank you for listening.